Hello, everyone, and welcome to today's live broadcast. Chimeric Antigen Receptors from Proof of Concept to Widely Applicable Clinical Research Solutions, presented by Dr. Dina Schneider. We're excited to bring you this educational web seminar presented by LabRoots and sponsored by Miltenyi Biotec. Miltenyi Biotec is a global provider of products and services that advance biomedical research and cellular therapy. The company's innovative tools support research at every level, from basic research to translational research to clinical application. This integrated portfolio enables scientists and clinicians to obtain, analyze, and utilize the cell. Miltenyi Biotec's technologies cover techniques of sample preparation, cell isolation, cell sorting, flow cytometry, cell culture, molecular analysis, and preclinical imaging. Their more than 25 years of expertise span research areas including immunology, stem cell biology, neuroscience, and cancer, and clinical research areas like hematology, graft engineering, and apheresis. The company also offers comprehensive scientific support, consultation, and expert training. To learn more about Miltenyi Biotec, visit www.miltenyibiotec.com. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Before we begin, I would like to remind everyone this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice you'll be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. Finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or you can use the Q&A button to let us know you're having a problem. Thank you. Now I'd like to introduce Dr. Schneider. Dr. Schneider holds a Ph.D. in Pharmacology and Toxicology from Michigan State University. Her doctoral thesis focused on pathways of B-cell activation and differentiation. Her postdoctoral work focused on cellular immune responses to viral infections in airway inflammatory diseases. In 2011, Dr. Schneider transitioned to industry, where she contributed to numerous projects in synthetic immunology, immunotherapy, and chimeric antigen receptor CAR T-cell-based therapy. Dr. Schneider leads the cell biology group at Lentigen Technology, a Miltenyi Biotec company. Her group focuses on preclinical development of novel CAR-based therapies and on the implementation of the CliniMACS Prodigy platform for preparation of cell-based therapeutics. Please join me in welcoming Dr. Schneider. I will now turn the presentation over to her.
Thank you, Judy, and hello, everyone. So today we will be speaking about CAR T-cells, and I would like to begin by giving you the overview of my today's presentation. So I am going to tell you a little bit about what CAR T-cells are, and I will show you how CAR T-cells are both an emerging technology and now 2 approved gene therapy products. And then we'll speak about viral vectors that are used or can be used to engineer T-cells for adoptive cell therapy. And then I will show you an example of a development of a CAR construct and its journey from R&D all the way to the clinic. And we'll speak about manufacturing of GMP-grade CAR T-cell products for clinical trials. I will give you an overview of CAR T clinical trials as currently practiced, and then we will conclude by saying a few words about CAR and NK cells. So with that, we will proceed. So what are CAR T cells? CAR T stands for Chimeric Antigen Receptor T cells, and these cells are generated through synthetic biology. So as you all probably know, an antibody depicted in the slide here in figure A and B is comprised of 2 heavy and 2 light chains. And each antibody has a region which is responsible for recognition of an antigen and antibody binding and that region is comprised of the variable heavy and the variable light fragments. Through synthetic biology, these fragments can be linked together to create what is called SCFV, single chain fragment variable. And those SCFV fragments are commonly used in the generation of CAR T cell receptors. So in the figure on the right is an example of use of SCFV-based binding domain in CAR structure. On the left of the figure, you can see depicted a tumor cell at the top and a T cell at the bottom. A receptor on the left is a CAR T receptor, and it's comprised of a binder, which is a variable heavy and light chain, that is linked by a hinge and transmembrane domain to intracellular signaling domains of the receptor, while the binder is responsible for recognition of a specific targeted epitope on tumor cell surface. The signaling domains are responsible for initiating an intracellular signaling cascade, which eventually causes T cell to become activated and kill the tumor cell. On the right, you can see a traditional T cell receptor, TCR. And as you can see, there is a lot of differences between a CAR and a TCR. I will point out just a few that will be important to distinguish CAR in our talk. One difference is in TCR signaling, the antigen has to be processed by tumor cell and presented in peptide MHC context to TCR, which is then activated and is activating the T cell. In our case, the presentation of the antigen is MHC independent. Therefore, it can target much broadly antigens present on tumors because it does not require recognition in the context of a specific MHC. So where did CAR T cells come from? This technology first emerged in 1989 and in a group at Weizmann Institute in Israel, directed by Dr. Zelig Eshar. And first publications were interesting but have not generated a lot of interest in the field. And it actually took a lot of years until this technology progressed from its first applications into actual clinical applications, as you can see in the slide here, from the first publication circa 1989 to 2010, when first clinical trials were done using CAR constructs. It took a long time. And this evolution included exchanging CAR targeting domains, exchanging CAR signaling domains, introducing a co-stimulatory domain here in the figure, the co-stimulatory domain depicted on the right for 2010 is CD28. And it took all these years to come from a general idea into actual applicable product that can be used in the clinic. So CARs are often referred to by their generation. What does that designation stand for? As you can see in this figure, each receptor, so on the left, depicted at the bottom left, is a T cell receptor complex. And this complex contains a CD3 zeta molecule. There are two CD3 zetas, which are the actual component of the T cell receptor complex that initiates the intracellular signaling and causes T cell to become activated. In first generation CARs, the CD3 zeta intracellular domain was fused to a transmembrane domain that was commonly from CD28 or CD8 molecule, which in turn was fused to anti-tumor targeting domain, the single chain FV derived domain, as I alluded to earlier. So in the first generation CARs, the only signaling domain present is the CD3 zeta. later on, it became understood that it's helpful for CAR T cells to have a second domain that helps with the signaling. And CD28 was the first co-stimulatory domain that was introduced. So if a CAR has a co-stimulatory domain, such as CD28, 4-1BB, or OX40, in addition to CD3 zeta signaling domain, that is a second-generation CAR. And even later on in additional development, there constructs were made that contain two signaling domains, for example, 4-1BB and CD28 together. And that is a third-generation CAR. So this is the nomenclature. Now, as you can imagine, now that this technology has been realized, we are in an era where we can view CAR construction as a CAR shop. Basically, the idea is that it's modular. You can select whichever you want CAR targeting domains, whichever you want linkers, whichever you want signaling domains, co-stimulatory domains, and combine them together to construct a CAR with the properties that you want. So you could select from, one type of construct that can be made is active CAR, which is comprised of a targeting domain, transmembrane domain, co-stimulatory domain, activation domain, or you can have constructs that have just CD3 zeta without the co-stimulatory domain, co-transduced together with constructs that only have co-stimulatory domain. In that case, you would split your signaling between two different molecules, and you would require engagement of both molecules in order to generate maximal T-cell activation. And as I will show you later, it might be important in some more sophisticated CAR designs. You could have tandem CARs where you have two targeting domains connected to each other, and so the CAR would be activating by binding either one of the two epitopes that are targeted. You can have inducible CARs, or you can have CARs that require a small molecule ligand for the CAR extracellular and intracellular portions to come together and initiate signaling. And all these innovations allow better control of CAR function. So one of the issues that stands in front of CAR field is CAR safety. Even though CAR has shown very promising results in the clinic, safety remains a major concern. And so over the years, there have been many approaches that have been tried in order to make CAR safer, or to better control the length of time throughout which the CAR is active. And so safety mechanisms that have been tried are safety by suicide, meaning CAR T cells can suicide when they are told to or when a treatment is applied, tagging CAR T cells, or transient expression of CAR T cells. So in the figure that I'm showing now, you have in A, B, and C three different methods for controlling CAR T cell presence. HSV-TK is a thymidine kinase, viral thymidine kinase derived molecule that is introduced into the T cell together with the CAR. And when a drug called ganciclovir is given to the patient, activation of this molecule makes T cell die by apoptosis. So that's one way that you could eliminate CAR T cells if they are causing toxicity in the patient. Another similar mechanism is depicted in panel B, and that's inducible caspase-9. And here, an addition of a soluble molecule, which is a dimerizer, allows dimerization of synthetic I-caspase-9 domains within the CAR T cell, which leads again to T cell apoptosis. The mechanism depicted in C is the truncated EGFR receptor. It's a cell surface receptor, which is truncated so that it does not have any signaling capacity, but it can be recognized by antibody cetuximab, which is a clinical grade antibody. And then the T cell can be targeted by elimination by ADC or ADCC mechanism. Again, that's a way of getting rid of unwanted T cells. And finally, generating CAR T cells by RNA electroporation results in transient expression of the CAR. And so the cells are only functional for the duration of CAR expression. The downside of this method, though, is that it would require multiple applications of CAR T cells for prolonged activity. So it might benefit in terms of safety, but the efficacy might be affected. Another way to control CAR function is by logical gates. So in the panel depicted here, you either have an inducible CAR, or a logic-gated CAR, or on-switch CAR, or a masked CAR. So each one of these mechanisms, again, allows you to activate the CAR in a more controlled fashion. With the inducible CAR, you would only activate the CAR if signal A but not signal B is present. And in this model, this only happens on tumor tissue, and that way CAR is not going to be active against normal tissue that might be expressing the same antigen that tumor is targeting. In logical gated CAR cells, activation of A and B is actually both is required for full activation of the CAR. In the on-switch CAR, there is an inducible ligand again, which is required for intracellular domain activation of the CAR. And masked CAR actually contains a domain that is masking the scFv binding domain, and when the CAR is present in tumor microenvironment, the proteases in the environment relieve the mask and allow the binding domain to bind. So these are all different methods which have been proposed for controlling CAR function. Another approach I want to talk about is the universal CAR. And this CAR is basically would be a product that would be generated from a universal donor and would be then being able to be given to any patient. And in order for that to happen, two modifications have to be done on such a CAR. One is the deletion of the endogenous TCR to prevent graft versus host disease, and another is the deletion of major histocompatibility complex genes to prevent immune rejection of the CAR by the host. So this approach is also being worked on. It's still in relatively early stages, but it's a very promising technology. And another technology I would like to mention is CRISPR-Cas9. It is impacting and will continue to impact cell engineering. And basically, as I told you, presently what we are doing, we are introducing the CAR molecule into a T cell that does contain its own TCR and does contain its own inhibitory receptors. And so it could be that because of that, the CAR function is not optimal. However, CRISPR technology allows to specifically target DNA insertions into a particular locus. And in that case, if, for example, we wanted to eliminate the endogenous TCR, it's possible to target CAR expression into the TCR locus. And that way, in one step, you would both remove the TCR and allow better CAR expression. So that is a very promising technology that is being worked on right now. Also, I want to mention to you that in order to select the best CARs for the job, another approach is possible. It's not only molecular engineering of the cell, it's also the selection of the relevant T cell subsets. And shown here is a publication by Dr. Gattinoni's group showing that by using stem cell memory T cells for CAR applications, it's possible to increase their functionality. Basically shown here are on the left in vivo model mice that did not get any treatment and got high level tumor burden by day 12. And then in the middle, a standard application of CAR cells, so they're not selected for any particular phenotype. And on the right, application of stem cell memory enriched CAR T cells, and you can see it results in more efficacious tumor elimination. So to summarize this part, there is a number of different emerging CAR technologies aiming to produce CARs that are better controlled, more functional, and safer. And the factors that matter are the selection of the initial cell substrate, such as stem cell memory or central memory cells, or CD4, CD8 positive T cells or virus-specific T cells. The other consideration is the gene vector. Is it just a CAR? Are there any additional factors added, such as soluble factors that increase CAR functionality? Next generation gated CARs, all the new innovations I talked to you about, to better control the time of CAR T cell functionality. And finally, target selection. So the targets that CAR T cell target are sometimes not unique for tumors. They're highly expressed on tumors, or they are specific to a lineage, such as in B-cell leukemias and lymphomas, and so loss of lineage is tolerable, even though it's not ideal. But one avenue that needs to be explored more in the future is finding targets that are very selective for the tumor and not present on normal tissue. That will also increase CAR safety. So now I would like to switch gears and talk about viral vectors for adoptive cell therapy. So retroviral vectors such as gamma retroviral vectors and lentiviral vectors are infectious and pathogenic. I'm sorry, the viruses are infectious and pathogenic. The viral vectors are based on the virus, but are modified in a way that they are replication deficient and can be used as a therapeutic. So depicted here is a viral life cycle. So when a virion infects a cell, it fuses with cell membrane and releases its genomic RNA, which is then transcribed to cDNA and integrated in the host genome as a provirus, and then transcribed and translated and assembled into virions which bud from the cell surface. That's the virus life cycle. Now, in the use of lentiviral vectors, several elements of the viral genome have been modified in a way that the vector is efficient in getting into the cell and delivering the DNA into host genome, but is incapable of replication and infection. So how is that achieved? The lentiviral genome depicted here between two LTRs is comprised of a number of structural proteins, and basically the GAG, the POL, and the ENV and the REV are the important proteins in the viral genome. So in order to generate viral vectors that are not able to propagate infection but are good for delivering DNA materials, a system has been generated called self-inactivated vector. And in this system, a number of steps have been taken to make it safe. So the first step was that the packaging vector, the transfer vector only contains the transgene material under a recombinant promoter, in this case PGK promoter, but not any of the structural genes that are required for viral life cycle and replication. That's the first precaution. Another precaution is that a portion of the LTR has been deleted so that the virus is unable to replicate on its own once it's in a provirus stage. In addition, the other components that are required to generate viral particles, GAG, POL, and REV, have been split into multiple plasmids. So in the 4-plasmid system case, there are 4 different plasmids. One is the plasmid containing the CAR sequence of interest, and a separate plasmid encoding the GAG and POL, and yet a separate plasmid encoding the REV sequence, and a separate plasmid encoding the envelope sequence. And that way, those plasmids, there's a very small chance that they would ever come together and recombine to form a functional virus again. And in fact, such case has never been seen in the clinic in all the history of application of lentiviral vectors. So CAR therapy requires production of lentiviral vectors at commercial scale. How can that be achieved? So in this diagram here is shown a schematic of the viral production process. So here we are starting with 4 plasmid system, and those plasmids are transfected into actinin 3 cells, which are the production line. And then viral particles are produced and secreted into culture supernatant. From there, they are isolated and purified and used for transduction of target cells. And then target cells acquire the gene of interest and express it. So at Lentigen, we have a fully developed GMP lentiviral vector manufacturing process to generate viral vectors for adoptive cell therapy. It starts from upstream culture to tangential flow filtration to chromatography to sterile filtration to bulk fill, and it produces highly pure lentiviral vector with robust functional titer. Now I would like to switch gears and tell you about a journey of one CAR from R&D to the clinic. So we are going to talk about an OR logical gate CAR that is targeting CD20 and CD19 antigens on tumor surface simultaneously. And it's intended for the treatment of CD19 positive and or CD20 positive hematologic malignancies. And the idea behind this approach is that by targeting 2 antigens on tumor surface simultaneously, we will be able to mitigate tumor antigen escape. So depicted here are 2 different CAR constructs. One that starts with a CD19 scFv linked to CD20 scFv, and in turn linked to CD8 transmembrane and hinge domain, and CD123, which is the same as 4-1BB domain, which is a co-activation domain, and CD38, a signaling domain. That's the structure of the CAR. That's one configuration. The other configuration is CD20 binding domain first, linked to CD19 domain, and then followed by CD8, CD137, and CD38 as before. And also, in addition to those tandem CAR versions, we have generated a single targeting control, targeting either CD19 or CD20. And depicted in the bottom panel is a flow cytometric analysis of CAR expression on cell surface. And in order to achieve this assessment, we use 3 different methods. One method is staining of CAR with protein L. Protein L binds to scFv light chains of the kappa type, and so it would recognize non-discriminately the CD19 and CD20 binding domains. And then on the right are depicted 2 specific detection methods that detect either CD19 binder or CD20 binder on the surface of the CAR-expressing cell only. And as you can see, in both cases, expression of the binders, expression of the CARs was pretty high. One thing to point out is for 1920 configuration, when we looked at CD20p, so expression of CD20 binder, we picked up a much less signal than we did for the 2019 construct. And that is a hint that in this configuration, the 20 binding is probably not optimal. So that will be important for future slides. So then we used the in vitro system for a killing assay to assess the potency of these CARs. And we used multiple cell lines that either expressed both CD19 and CD20, such as Raji and Nalm. And as you can see, when both antigens were expressed, we got specific killing using CARs expressing the recombinant antigen. 293 T cells are a CD19, CD20 negative line, so there is no killing in this line. The RAH clone we have is CD20 negative, and as you can see again, the CARs that express either CD19 only or CD19 and 20 were able to kill this line, but not CAR expressing CD20 only. Again, it's an indication of specificity. And in the top row shown are K562 cells, which are again CD19, 20 negative, so there is no specific killing, or K562 cells that specifically express CD19 or CD20, and again, the killing was antigen specific. So we see that our CARs are specific to the antigen they are targeting. The next figure I'm showing is cytokine release assay for CAR cells co-incubated with Raji tumor line. And as you can see here, whereas the single CARs targeting CD19 both induced cytokines interferon gamma, IL-2, TNF-alpha, and GM-CSF, the CD20 single targeting CAR induced much greater levels of cytokines. And by contrast, the tandem CARs produce cytokines at intermediate level, which might be optimal for clinical applications. And then we proceeded to test our CARs in vivo model in Raji mice, in Raji tumor model in NSG mice. And as you can see here, tandem CARs were able to eliminate tumor efficiently. So then we wanted to look at the mechanism of tumor antigen escape and how our CARs behave in that regard. And the reason we are concerned with this issue is that in the clinic in CD19 trials, fairly large amount of patients had relapsed with CD19 negative tumors. And so it's important to understand this phenomenon and to find ways to mitigate that. So what we found when we co-incubated our CARs with Raji cells or with Nalm cells, we found that CARs targeting CD19 alone downregulated CD19 surface expression on the tumor, similarly to what has been seen in the clinic. However, the tandem CARs preserved much greater levels of CD19 expression and also allowed the CD20 levels to be preserved at greater levels. So that makes the tandem CAR a more favorable construct to target both antigens with low risk of losing them. And then finally, to better differentiate between our CARs, we performed another in vivo experiment in which mice were bearing tumors with very high burden. So we allowed the tumors to grow in the mice for much longer periods of time. And we tested either the 2019 tandem CAR or a mixture of CD19 CAR cells and CD20 CAR cells, noted here as 19A plus 20A or 19B plus 20A, or a single application of either CD20 CAR or CD19 CAR. And what we found out is that the best profile in terms of tumor clearance and mice survival was obtained for the 2019 tandem CAR. As you can see here, all 6 mice survived and the tumor has been greatly controlled, greatly reduced. For 19A and 19B, 5 out of 6 mice survived, and tumor was still present. For all the groups that contained CD20 CAR, either by itself or in combination with a CD19 CAR, 2 mice in each group where the tumor control was very good for 2 mice in each group. However, the rest of the mice were found dead, and that is probably due to high toxicity of the treatment, which might mimic cytokine release syndrome in people. So what we found out is that the 2019 CAR is both effective in tumor elimination and has a more favorable toxicity profile in this NSG mouse model. So we work with a very talented group of investigators at the Medical College of Wisconsin, led by Dr. Nirav Shah, and we are happy to report that now a clinical trial has been opened for the 2019 CARs in patients with relapsed or refractory B-cell malignancies, and we are very excited about that. And now I would like to speak about the manufacture of CAR T cells. So as you probably can imagine, there are multiple challenges for ex vivo cell processing of cell and gene therapy products. And curing manufacturing protocols can be characterized by complex processes which have low standardization and low reproducibility and high levels of manual operations. There are many open processing steps, and there is a high risk of quality events, and overall cost of goods is relatively high because of all of these factors. So another way to generate CAR T cells is to have an integrated process. So the steps required in CAR T generation is blood leukapheresis, cell selection, cell activation, cell transduction, cell expansion, cell product formulation, and finally administration to the patient. And as I have already said, these steps are labor intensive and prone to error and variability and require use of multiple types of equipment. So Miltenyi Biotec has developed an instrument which offers an automated cell processing solution called CliniMACS Prodigy. And this instrument performs integrated cell processing from starting material to final cellular product, including sample preparation, cell washing, density gradient separation, max cell separation, cell activation, genetic modification, cell culture, and final product formulation. And therefore, it enables a complex process to be fully automated and controlled. And it's done in a closed single-use tubing set system. So the manufacturing and distribution of autologous ex vivo cellular products is complex and is hard to scale if early development stages such as IND and phase 1 clinical trials may require only a small amount of patients to be enrolled. As the studies proceed to phase 2 and phase 3 and finally commercialization, much larger amount of patients are enrolled and much larger scale is required for the product manufacturing. And it has been already noted that the challenge of scale is a bottleneck in CAR T therapies today. So there are two different models for manufacturing CAR T cells. One is the centralized model and another is point of care model. In the centralized model, the patient is being treated at a hospital and his blood is being collected at the hospital and then is transported to a centralized location where cell processing occurs, and then it's being transported back into the hospital where the patient is and administered. That's one model. In the point of care model, the facility where the cells are being produced is found in the proximity to the hospital, and this allows more streamlined and less logistically laborious processing of cells. So today, for example, the manufacturing of CTL19 product, is a strong demonstration of centralized processing. The patient is referred to the trial, the cells are obtained, and then the cells are transferred, usually flown into a centralized processing facility in Morris Plains, New Jersey, and processed and then flown back, and then delivered at the hospital, and then the patient receives therapy. And then the other option, alternative to that, is point of care model, where everything would be done at the same location pretty much, and there are less processing steps, less handling. So now I would like to say a few words about CAR-T clinical trials, and by just giving you an example of a process through which cells are prepared for a trial. So in the typical trials, the lymphocytes would be collected from the patient about 11 days prior to CAR-T administration. And during that time, T cells would be activated, expanded, transduced with a CAR, and then expanded, formulated, and finally given to the patient. And usually prior to CAR-T administration, chemotherapy is given, which usually consists of fludarabine and cyclophosphamide. So the application of chemotherapy is important because lymphodepletion is very important for a maximal CAR-T function. And that is for a number of reasons. The lymphodepletion makes space for CAR-T cells to engraft through several mechanisms. One is by increasing the level of free cytokines IL-7 and IL-15 in the body that are available to stimulate CAR-T cells. Another mechanism is by removing regulatory T cells, which might be inhibitory to CAR-T function. And finally, by altering tumor and bone marrow microenvironment, which again potentiates the functionality of CAR-T cells. So among the ongoing clinical trials, the most prominent one probably is Novartis CTL019 trial, ELIANA. It's a trial for pediatric and young adults with relapsed or refractory B-cell acute lymphoblastic leukemia. And it's the basis of BLA. So far, 82% of complete responses have been reported. This is a very good result. And the pivotal data is supported by CTL019 multi-center trial called ENSIGN, as well as earlier single-site trial. So this trial showed relapse-free survival rate at 6 months of 60%. It enrolled in 25 centers across Europe, United States, Canada, Australia, and Japan. About 48% of patients had grade 3 or 4 cytokine release syndrome, but there were no deaths in this trial. And 15% of patients had grade 3 neurological and psychiatric events, including encephalopathy and delirium. That's one of the side effects that is known to be associated with CAR treatment. And the overall response in the adults was 47% in diffuse large B-cell lymphoma at 3 months, and it was 73% in follicular lymphoma. Another trial for product KT-C19 has been conducted by Kite. Actually, it's a series of trials called ZUMA. And again, here they are both. Some of them are phase 1 and some phase 2. Again, it showed very good overall responses at 3 months, and for some endpoints, the data at 6 and 9 months was released, and it's also pretty good. So here is just an illustration of a patient that has been treated in the ZUMA trial, and as you can see, there is a response to treatment. So Juno is another company that is actively conducting CAR T trials. There are several trials ongoing. JCAR 014 is being conducted at Fred Hutchinson Cancer Center, and it's a CAR-19 with TGFR tag generated by lentiviral transduction. Another trial, JCAR 15, was unfortunately terminated. And another ongoing trial is JCAR 17, conducted at Seattle Children's Hospital. The construct also has a TGFR tag. And in this trial, a defined combination of CD4 and CD8 cells, 1-to-1 combination is used, and the cells are transduced by lentiviral vectors. And in this trial, the results were also very promising. So cyclophosphamide-only conditioning produced 8% complete responses and 50% overall response. Cyclophosphamide with fludarabine conditioning produced 50% CRs and 72% overall response rate. And at maximal tolerated dose, the combination chemotherapy produced 82% overall response rate. So now I want to say a few words about NK cells. So NKs are another type of immune cells which are capable of killing tumors, and the possibility of using NKs as CAR cells is being investigated. This is a relatively new emerging area of study, but there are several ongoing clinical trials at this time. One is sponsored by MD Anderson Cancer Center, another by Baylor College of Medicine, and there is a couple of trials in China. So this is a promising new area. So to summarize, CAR T cells continue to show great potential in the treatment of cancer. Further improvements to the efficacy and safety of CAR therapy are being developed. The use of lentiviral vectors and automated integrated clinical manufacturing protocols offer solutions for streamlined CAR production. And multiple clinical trials are underway for CAR T products. It's actually, the count is in the hundreds. And there are several CAR and K trials ongoing as well. And CAR and Ks are emerging as a new approach in CAR-based therapy. So with that, I will conclude, and I will be happy to take your questions.
Well, thank you, Dr. Schneider, for that informative presentation. It's time for Q&A. If you have a question you'd like to ask Dr. Schneider, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your question into the box that appears on your screen and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, you mentioned in your presentation that T cells were selected using anti-CD4 and anti-CD8 beads. Are there any other reagents available for the selection of T cells?
Yes, in fact, CD62L reagent is available as well for selecting CD62L positive cells. And use of that reagent may, in fact, result in a product that has a higher component of central memory T cells, which, as I have shown you, are predicted to be more functional in vivo.
What are the side effects associated with CAR T treatment?
Yes, so currently there are two major side effects that have been described in multiple trials. One is cytokine release syndrome. And this side effect is basically typical of a class of drugs. It's typical of monoclonal antibodies and also is seen with CAR T cells. And it usually manifests as fever, nausea, chills, hypotension, and dyspnea, and in severe form it might even be fatal. But the good news is that there is a drug that can be used to minimize cytokine release syndrome. It's an IL-6 receptor antagonist called tocilizumab. and it has been able to manage cytokine release syndrome in many patients. Another type of toxicity is a neurologic toxicity, and it's being investigated currently. And what is known to date is that both the CRS, cytokine release syndrome, and neurologic toxicity occur more when either high tumor burden is present or very high dose of CAR T cells is being given, or the patients had a prior history of central nervous system problems. And so all of these observations will help in the future to determine which patients are more at risk and to give appropriate doses of CAR T cells to prevent similar toxicities from occurring.
Is it possible to culture T cells on CliniMACS Prodigy with other cytokines rather than IL-2?
Yes, indeed. One option that comes to mind is the use of IL-7 and IL-15. And this may result in even fitter, better CAR T cells.
Thank you. I would like to once again thank Dr. Schneider for her presentation. Do you have any final comments?
Yes, I would like to thank LabRoots, the organizers, for giving us the chance to present. And I would like to thank all my colleagues at Lentigen Technology and all my colleagues at Miltenyi Biotec for being part of this wonderful work. And I feel very fortunate to be a part of that.
Well, thank you once again. I would also like to thank our sponsor, Miltenyi Biotec, for making today's educational webcast possible. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May of 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That is all for now. Thank you for joining us. We hope to see you again soon. Goodbye.